Good morning, everyone. Thank you for your time and interest. So Vivet Therapeutics is a liver-targeted metabolic gene therapy company, and we're really focusing on rare inherited metabolic disorders. The focus of the company is to develop long-lasting gene therapy for a specific metabolic disorder, notably Wilson disease. I will detail more. So we have, effectively, we're funded by blue chip investors. We have a significant collaboration with CIMA, I will detail, and we're really willing to constitute and look at the AAV technology specifically because you need to have a significant level of expression. And also we have some innovative platform looking at how we can optimize the target of liver gene therapy. The company is back with an experienced leadership team, and you can see the type of investors we have with a significant network of expertise as well. So the way we have constructed the company is really to build a disruptive development engine because this collaboration is really bringing to us a strong translational expertise with three core competencies with molecular biology, disease biology, and immunology under the responsibility of Gloria Gonzalez, our Chief Scientific Officer and co-founder. And we're really controlling our destiny in a way that we can complement the key platform regarding the needs of bioanalytical, large animal facility, including non-human primate, imaging department, which are of significant importance on the gene therapy field. The company pipeline we have built is really focusing on liver metabolic disorder with Wilson disease with an ongoing clinical program. We're also working on a NAB depleting strategy on how to eventually treat NAB-positive subjects, but also we have multiple assets as an ongoing IND-enabling study with an indication called CTX. I will give a few details. But also we built a platform on PFIC disorders, progressive familial intracholestasis disorders, citrullinemia, also PKU. And we're really willing to understand how we can improve the damaged liver regeneration platform because some of these patients we're targeting have liver fibrosis, and we know that it could be a significant limiting factor on how AAV can actually transduce the liver. So Wilson disease, in a nutshell, it's a rare debilitating disease where you have some significant accumulation of copper. And this copper accumulation is working through a small protein called ATP7B, where you have a significant deficiency. And it has two major roles. One role is actually to be able to excrete the free copper from the cells and eliminate through the bile duct, but also the increase in ceruloplasmin, which is actually transporting iron and copper. And when you have no ceruloplasmin increase, specifically in terms of functionality and specific activity over copper, you accumulate free copper, and that can have neurological and hepatic damages. So it's a multi-organ target toxicity. The current treatment are very basic. They are chelators, which actually pump the excessive copper in the bloodstream, but not from the tissues. What we were striking recently, we actually look at this disease from a real-world data, and we observed that these patients actually have a higher and earlier mortality with a lot of consistency between different countries from Sweden, UK, France, US, Germany, and also South Korea and Japan. So despite treatment, a lot of patients continue to have damages and progression, notably neurological, about a fourth of the patients. So there's a high unmet medical needs because this disease is diagnosed rather lately. If you look at real-world data compared to some communications, patients have continuous clinical manifestation despite being on standard of care. The mortality is important, and up to 10 patients can receive a liver transplantation. So we have built this opportunity with Gloria Gonzalez through multiple papers and highly peer-reviewed journals. This program received orphan drug designation, fast track designation, and we have a key phase 1/2 ongoing with key results in H2 24. But what is interesting is to look at how we built this gene therapy program, VTX801. The wild-type gene of ATP7B is pretty large. It slightly exceeds 4.7 kilobase. So there was some mini ATP7B optimization, keeping the 4, 5, and 6 metal binding domain. And in this independent work clearly showed that the ATP7B mini is superior to the wild type, expressing the full functionality. The key question in copper metabolism to answer is, where is the copper going? It's like an excretion mass balance study, basically. In normal healthy volunteers, like or anybody, has an excretion about 8 to 10% in the biliary excretion and feces, but you can see in any other, either heterozygous, have a slight delay, but the other patients who have gene mutations actually have urinary copper mostly. So what we did is we use a tracer, and this is the importance of the imaging technology, and looking at the copper-64 elimination in the feces, you can see that the gene therapy restore full functionality in the dose response, but also restore the ceruloplasmin full expression. And that actually is leading to neurological protection, but also elimination of copper directly from the liver. So we're pumping out the excessive copper in the liver, but also a full 100% survival. And that has been shown with PET scan analysis. We have published, and you can see the very significant excretion from the 5e12 to 1.5e13 Vg per kilogram dose, and in a dose response, a full restoration of the metabolic. So what we're applying currently in the phase 1-2 dose escalating, we're applying this pure technology where we're going to measure the baseline of the copper-64 elimination, then we give the gene therapy, and we measure over 12 weeks and 36 weeks, and we have a 5 years follow-up the effect of a dose range on copper-64, which is a very reliable biomarker, because copper homeostasis biomarker are pretty viable. So here we really want to show the full restoration. And this data will be available by H2 24. On the other platforms, we're also working on cerebrotendinous xanthomatosis. It's a rare autosomal recessive genetic disorder that affects metabolites, I would say, fats known as cholesterol. It's a very interesting disease in the sense that it's diagnosed very lately, like most in 30 to 40 years as a mean, and it targets cirrhosis, cirrhotic, it targets the cataract tendon, but most importantly, neurological damages. So it's a complex journey for the patients, and what we've been able to show, again with the CIMA collaboration, is that we've been able to show that the full restoration of the functionality, but also a significant decrease in toxic metabolite at a relatively low dose. And this program is currently under IND-enabling studies, and we've been granted a significant grant from the French Public Bank of Investment this year. So we believe this program can be in clinic by late 25. We all understand the issue of neutralizing antibody, and this issue of neutralizing antibody is very significant because between let's say 15 to 50, 60% of the subjects can have actually pre-existing antibodies against all kinds of different AAV. We know there's a significant cross-reactivity. But when you have no pre-existing activity, you can be directly included in any gene therapy trials, but then you get a significant immune response. So what we've been working on is developing a product which can actually cleave the antibody. We've been publishing this data in the monkey, but also in mice, and we can see that we can extract and see the full restoration of the expression. However, in the monkey, we know it's not the full expression we can have, but we're currently running a phase 1 healthy volunteer NAP-positive subjects when we can detect the effect on various neutralizing antibodies of this product and create windows of opportunity of treatment. So in a nutshell, the company will be looking at the end of these results with Wilson disease, CTX, and this VTX-PID program on NAbs for future financing to initiate the phase 3 program, the platform and expansion with key results from Gateway. We will also continue to value the expression of this CTX program, but importantly also the platform on PFIC disorders and liver fibrosis. With this modular platform of NAbs, which we believe is important to expand this high unmet medical need because PFIC disorder generally lead to liver transplantation, and we have some GMP manufacturing collaboration to ensure the optimization of the full MT, commercial scale, and cost of goods from the beginning. Thank you for your attention. I'm happy to answer any questions. Otherwise, I'll be available all along this presentation, and thanks to the team for all this hard work.